So now we're going to look at the process of what is called hematopoiesis. And hematopoiesis refers to how we make blood cells. So it's poiesis means to make, basically. Um, so most of our cellular components of blood are made in the red bone marrow. So you'll see that the only place as adult humans we make red blood cells is in the red bone marrow. The only place we make platelets is in the red bone marrow. And most of our white blood cells are in the red bone marrow. But we can make a few of the white cells in the lymph tissue. And in terms of the lymph tissue, there are only two types that we can do, and that's lymphocytes and then monocytes. And so we can make those in the lymph tissue. One of the things that we often notice on artistic renditions of things, especially in science, is there's often mistakes. And here's a mistake that you probably just want to point out. If we look right here, this indicates the red bone marrow, right? The red bone marrow is not that, right? This is actually the yellow marrow. This is the medullary cavity. And so this is where the yellow marrow would be. This would be fat. The red marrow is up here at the ends of long bones within the spongy bone, and we'll see the red marrow there. But the bottom line is the red bone marrow produces most of the blood components. So as I said previously, that we're going to look at the process of what's called hematopoiesis, which just means making red blood cells. All of our red blood cells are derived from what we call a single progenitor, a single precursor cell. And that's a very important one. It's called a pluripotent hematopoietic stem cell. So I'll circle it. Pluripotent means like plural, many, potent, become, hemato, blood, and poietic, make. So basically, and a stem cell is a cell that can turn into other types of cells. So the pluripotent hematopoietic stem cell is the cell that can become the other types of blood cells. And so if you look right here, if it undergoes the right chemical signals, which we'll talk about, it can become eventually, go all the way down, a red blood cell. It can become platelets. It can become all 5 of our different white cells as well. Okay, so we're not going to worry too much about all of the nitty-gritty details in these, but we'll talk about the general overview and some of the different aspects of it. But the key cell, again, is this stem cell that's called a pluripotent hematopoietic stem cell. And this is the one that's going to be responsible for making all of our other blood cells. So let's look at the way we produce a red blood cell. Remember, a red blood cell scientifically is known as an erythrocyte. Erythro means red, cyte means cell. So again, we start with that pluripotent hematopoietic stem cell that we talked about before, and it gets a chemical signal that says, hey, go make a red blood cell. That chemical signal is called erythropoietin. Erythro, again, red, poietin means make, so basically that's hormone name says make a red blood cell. Oftentimes in different circles, medical circles, as well as athletes who sometimes use erythropoietin to cheat, they call it EPO. It's produced in the kidney, and its production is induced by low oxygen, or what we call hypoxia. Hypoxia is the way you pronounce it, but hypo, low, and then oxia, oxygen. So hypoxia. So hypoxia will stimulate the production of red blood cells, right? And so the way that happens is that is sensed in the kidney, and basically we produce more erythropoietin that says take more of these stem cells and eventually make a red blood cell. Now, if you look at the Olympic Training Center, there's a couple in the United States, but the primary one is up in Colorado. And one of the reasons why we have the Olympic Training Center in Colorado up in the mountains is the high altitude training camps. And what happens is when you live at high altitude, you stimulate the production of erythropoietin. And so then you make more red blood cells because you're hypoxic. And as an athlete, if you can have more red blood cells, you can carry more oxygen. And we saw the advantages of metabolic things with oxygen, making much more ATP from, say, a glucose molecule compared to anaerobic glycolysis. And in fact, we can't even break down lipids without oxygen. So having more red blood cells is actually an advantage in most cases. So there are advantages to having synthetic EPO, and that stimulates red blood cell production. And we could give that to patients that were undergoing chemotherapy where we knew they were going to become anemic, or people that had severe anemia and things like that. So what happened, though, is athletes figured that, hey, you know what, if I take this synthetic erythropoietin and take it as an injectable, then I don't have to train at altitude, right? No training necessary. And I can make more red blood cells to compete better. It's, a very hard thing to detect, although, we have been able to detect it in humans and even in racehorses, it's very difficult to detect. And what it does is it allows people or horses or other athletes that take it to compete better and give them an unfair advantage. Ideally, to try to level the playing field, you'd be able to test for it. And up to a certain point, we weren't able to test for it. Before the 2002 Salt Lake City Olympics, we could not test for erythropoietin. There was no test to look at that. They would tell athletes if their hematocrit was excessively high. Remember, your hematocrit is your packed red cell volume. And so if you increase the number of red cells, your hematocrit's going to get bigger. It also depends on the size of the red cells. But if you increase the number of normal sized red blood cells, your hematocrit's going to get larger. The higher your hematocrit is, the more viscosity of blood you're going to have, but you're going to be able to carry more oxygen. So in the Salt Lake City Olympics in 2002, they actually figured out how to test for the synthetic erythropoietin and didn't tell anybody. And so what they were doing is they were doing the tests and not looking at it. And not letting the athletes know. So here's the Johan Muleg, who won 2 gold medals initially, and then won a 3rd gold medal. In the first 2 gold medals, he was fine, but for the 3rd gold medal, he was actually taking synthetic erythropoietin. And so there was this big controversy because they said, well, if he cheated, even if he was only shown to cheat the one time, shouldn't we take all 3 gold medals away? And eventually they took all the gold medals away from him for cheating. And in fact, there's been, that year there was more than 100 athletes that tested positive for blood doping. So, one of the things they try to do to try to discourage blood doping, especially if athletes have a potential drug that they can use that's not detectable now with any of the tests we have, is they will freeze samples from athletes from Olympics and things like that. A couple years ago, a member of the Summer Olympic relay team that won the gold medal with Usain Bolt uh, he wasn't tested. He didn't test positive for a drug, but one of the relay members did. And so they took away the gold medal from him. And since he was part of the relay, they took the gold medal away from all the athletes that participated in that relay. And that was a couple years after the Olympics, where they continued to test for things where they thought athletes may have been cheating. Now, not surprisingly, we have a similar named hormone called thrombopoietin. So remember, the last one was called erythropoietin. It's erythro because it made red cells. So thrombopoietin, right, makes platelets. So you can see them down here, platelets. We start with the same hematopoietic pluripotent stem cell. It goes through a series of steps, eventually turns into what they call a megakaryocyte. Mega, large, karyo refers to kernel or nucleus. It's got all this stuff inside the cell, basically. And then that breaks up into a whole bunch of pieces that become a platelet, or actually many, many platelets. So thrombopoietin stimulates the production of platelets. So it's the same starting cell that just gets a different chemical signal that says, hey, make some platelets. So thrombopoietin helps to make platelets. Now, white cells are a little bit different. So the leukocytes are a little bit different, and they involve two chemical signals. And the reason why is we have one type of red blood cell for the most part. Now, a red blood cell that has a nucleus is called a reticulocyte, and it's really not a mature red blood cell, so technically it's not a red blood cell anyway. And there's, platelets become, activated or be inactive, but they're still a platelet. But the white blood cells have a number of different subtypes. And so we need actually two chemical signals then in order to produce white cells. So again, we start with that pluripotent hematopoietic stem cell we've seen a couple times. But this time we have two chemical signals. The first chemical signal is called leukopoietin. And leukopoietin is basically the generic chemical signal that says make a white cell. It's not any different than the other two ones we've looked at. Leuko means white, right? It's the scientific name for white blood cells. So erythropoietin, right, makes red cells. Thrombopoietin makes platelets, and leukopoietin makes white blood cells. However, making a white blood cell is not sufficient, because then we take that white cell and turn it in to a specific subtype of a white cell. And so we'll learn later these, but our white cells are leukocytes. There are 5 major types: neutrophils, monocytes, basophils, eosinophils, and lymphocytes. And so in order to signal, hey, now become one of these specific subtypes, we need what's called the colony stimulating factor. So colony refers to the different subtypes, and stimulating obviously would be to make them, and factor is the chemical. So there's a 2 chemical signal setup for white cells. You need the leukopoietin that generally says, hey, make a white cell, and then you need the colony stimulating factor that says what specific type of white cell to make. Now, what's interesting is, we talked about red blood cells, and we said red blood cells have like 5 million. So 5, 0, 0, 0, 0, 0. 5 million red cells in a microliter of blood. While the white blood cells, let's just kind of go with the lower limit, they have 5,000. So if you look at that and you say, okay, how many red cells are produced in relation to white cells within the red bone marrow? You would say, oh, look, there's literally, 100,000, 10,000 times, 1,000 times. I could do math in my head. 1,000 times greater number of red cells than there are white cells. Therefore, we must make more of them. And the answer is no. So there's one other factor that's important. It's not only how many there are, but it's how long do they last. And, the red blood cells on average last 3 to 4 months. So, if you made a red cell, today. When I'm making this video, it's the end of March. So I have all of April, all of May, all of June, and probably past July 4th, where this red blood cell will last. And so I don't need to replace that one for a while. White blood cells, though, have a very disparate lifespan. They can last, in some cases, seconds, usually in some of them minutes, maybe hours, and some technically or I guess realistically last your entire lifetime. But in terms of white blood cells, there's one type of white blood cell called a neutrophil. Neutrophils make up roughly 2/3 your white cells, and they don't last very long. In many cases, if you have an active infection, so you can see the neutrophil right here in the middle, right? If you have an active infection, the neutrophil gets released into the bloodstream. It goes to a site of infection and attacks whatever's making you sick, engulfs it through phagocytosis, and then destroys it. But in the process, destroys itself. So neutrophils don't last very long, and they make up the vast majority of white blood cells. So we're constantly replacing neutrophils. So even though there's significantly fewer white cells than there are red cells, the actual production in the bone marrow is 75% of the cells made in the bone marrow are white blood cells, and only 25% are red blood cells because of that. So you don't just look at the total number, you look at the lifespan. And that's ignoring the platelets, but if you look right here, you can see this one big, huge megakaryocyte, right? So I'll get rid of these. This one big megakaryocyte, right, becomes all these little platelets within the bloodstream. So this kind of just shows the different maturation processes of different things. Here you can see a red blood cell, right, that has a nucleus, then it loses its nucleus, and then it enters the bloodstream. And you can kind of see the different types of other white cells that are here. So this is the red bone marrow that makes most of our cells. So in summary, for the red blood cell production, it's called hematopoiesis, or sometimes known as hemopoiesis. We start with the pluripotent hemopoietic stem cell, and it become any of the different formed elements. The red blood cells and platelets need one signal. The red blood cells need erythropoietin. The platelets, which scientific name is thrombocytes, need thrombopoietin, while the white blood cells need two signals. It needs leukopoietin and then the colony stimulating factor. Leukopoietin determines we need to make a white cell, and the colony stimulating factor, sometimes known as CSF, determines the particular type of one. Also, we mentioned two of the white cells can be made in the lymph tissue. That would be a lymphocyte and a monocyte, although most are made in the red bone marrow as well. And then the production level, we talked about how we make 75% of our cells are white cells, and only 25% are red cells because of the difference between their lifespan. So even though we have many, many, many more red cells than white cells, red cells last much, much longer than white cells. So we need to make more white cells to replace the ones that are dying off at a lot higher rate.